Welcome back to Day 2 of Advances in Drug Discovery and Development 2023. I'm George Hammond, Conference Producer at Technology Networks, and I'm here to introduce our first talk of the day, Screening in Organoids and Complex 3D Assays Using High-Content Imaging. I'm incredibly pleased to welcome Dr. Michiel Fokkelman as our first speaker. Dr. Fokkelman is the head of high-throughput screening at Crown Bioscience, Netherlands. He completed his degree in tumor cell biology at Leiden University, in which he applied lab automation and high-content imaging for multiple screening projects. His research focused on drug discovery in a broad range of therapeutic areas and assay modalities, applying different detection technologies for HTS and profiling projects in both small molecule and large molecule domain. A very warm welcome to you, Dr. Fokkelman. Following the presentation, we will have a Q&A session, and we welcome any questions you may have. Please ask questions using the Q&A system to the right-hand side of the video player. And now, without further ado, I would like to hand over to Dr. Fokkelman to begin his presentation.
Well, thank you very much for the kind introduction, and of course, I'm very happy to join this symposium. We'll quickly get into our platform. So today, I would like to talk about our in vitro platform that we've developed, which is basically built around three technologies that have really emerged in the past decade. So one of those is 3D cell culture, and we use that in combination with cell lines, organoids, and primary tissues. The second technology that really matured is imaging. So we use high throughput and high content imaging to study those tissue models or cell models. And then finally, we've established this whole platform using lab automation, so that we're also able to build a consistent repertoire of assays and protocols to keep on producing high quality data and to get really consistent results for these studies. To start with our in vitro platform, let's first look at our models. So we at Crown Bioscience have a very large collection of models. This ranges from well-known cancer cell lines, so the ones that you will find in most of the labs. As soon as you culture these already in 3D, you can already see that they start behaving differently and that they start to show their natural biology or their own characteristics. And when we take these cell models and make them actually more complex, so when we use, for instance, the PDX-derived organoids or even patient-derived organoids, we're slowly getting closer to the patient situation and using cell models that are completely representative of the patients or the tissues that we're trying to study. And of course, interestingly for PDX-derived organoids is that we have these matched with our PDX models, so you can move studies from in vitro to in vivo. But also for the PDO models is that we have matched pairs, for instance, between the healthy and the normal tissue together with the tumor tissue. So you have a healthy organoid and a tumor organoid. And we have matched pairs with organoids coming from primary, but also metastatic areas. So you can also study the disease progression using these organoid technologies. And of course, what's very nice about these organoids is that they are really nicely showing the not only the heterogeneity of the patient tumor, but they're completely representing the genetic and morphological changes that are present in that particular patient. So as a set of samples and models, they are very representative of the patients that we're trying to help with the research in oncology and in immuno-oncology. And then finally, making it even more complex, we have a particular platform where we use patient tissues directly from the hospital or from the tissue provider, which is our ex vivo patient tissue platform. And there we completely preserve the native tumor microenvironment and use that for testing potential new drugs. If we look a little bit closer at the organoid models, which is one of our biggest assets at this moment in vitro, this is also one of the things that have really grown in the past few years with the technology being developed by the Hubrecht. When we've been in parallel with them, have been working to expand the biobank of these patient-derived organoids And this has been growing ever since, and it's currently out more than 500 models. But they represent all the different types of tissues that or cancer types that we can find in the patients. And the nice thing with these organoids is that they really reflect and maintain the genomic and mutational profiles of the patient, and that we can keep on expanding these without the models changing their behavior and their genomic backgrounds as well. So they nicely reflect the patient population that we're trying to study and that we're trying to address in the drug search for cancer. Of course, with so many models, we also need to make sure that the data that we have for them is easily accessible and also searchable. So we have databases online, they're on our website, and they allow you to browse all the models that we have in our collection, and allows you to review data from the microscopes that we have in our labs, but also the responses to standard of care drugs and other growth characteristics. And we even have expression data and mutational data available as well. So it allows you to search the organoid models, but also the cell lines that we have, and make a selection in case you're looking to do a study with us, that you pick the model that's most suited for your research question, or even a panel of models, which can even be more representative in that case. So with these models, we then have that second pillar that we talked about in our in vitro platform. That's a high content imaging. It's one of our favorite things to do, and it's very simple, or it has a very simple reason. It's because it just provides so much information. When we use high content imaging, we always capture really nice images. We can directly see what cells and organoids are doing, how they're growing, how they're developing, but also how they respond to particular treatments and therapies. And thereby it helps us better understand how a complex disease is developing, or how its a potential treatment is targeting that disease. So in contrast to the traditional cell-based assays and the earlier 3D assays, which all relied on lysis of the sample or analyzing the supernatant, with high content imaging, we can actually capture all the individual cells in the whole 3D environment. We can actually thereby analyze also the response of the entire population of organoids within that well, and also within the whole 3D structure. And as an example here, we have some pictures that showed where we looked at the internalization of cetuximab versus an EGFR targeting antibody. We can see that indeed the cetuximab is nicely retained on the outside of the organoids, whereas the test antibody is showing internalization. So it's one of those examples of rich information that you can extract using high content imaging. And indeed, combining this with the 3D biology, we have a very physiologically relevant model to study this internalization of this particular drug. So we then combine those, that large collection of models and the high content imaging. How do we get to a platform? Well, this has been some years in the making, and I've been very glad to help here, is that we've set up this whole automated platform for high content screening and for these assays in 3D. So it's all built around using liquid handling, using lab automation to standardize as many procedures as possible. And by having it completely standardized, we are able to create a lot of consistency from experiment to experiment, and able to ensure that we always get the same, at least technical performance. And then when we know that the models that we work with are so robust, we can also guarantee reliable and reproducible results. So this whole platform is based on seeding organoids or tumor cell lines or tissues directly into 384 well format plates using hydrogel, and then they are cultured in 3D. We can add the drugs directly in 3D. We can add immune cells for IO assays. There's quite some options in that sense. And then depending on the design of the study, we can have a shorter assay of 4 to 7 days. But if needed, we can also go a lot longer, use a media refresh, and then have extended studies. And ultimately, by fixing and staining the samples and subsequently using the optical sectioning to capture the whole 3D cell culture, we can then analyze the response of all the cells in that same 3D environment. So we have a customized pipeline with our own proprietary software that allows us to then analyze all of the cells and all of the organoids that are captured in that whole 3D environment, and then calculate more than 300 of phenotypic and morphological readouts and features to start studying their response to the treatment. And with the data, we're then able to build profiles. It could be phenotypic, could be more simple, representative of viability, but allows us to build those profiles and use that to classify or understand how drugs are working. So we can draw the standard concentration response curve, calculate an IC50, or we can also look at very specific features that tell us how particular drugs are working or targeting, like the example with the cetuximab and the internalization of that antibody. But also with for instance, organoids, if we have like cystic models, we can also look at the epithelium formation and how big the cyst size is, or the wall of that cyst is. So there's a few, there's quite a few things that we can analyze using high content and using the rich data from high content. And by having this whole platform built on a set of liquid handlers, we can actually run this quite consistently, not only in the lab here in Leiden, but also in our lab in Utrecht. And it's almost agnostic to the liquid handler itself. It gives us a lot of flexibility to run these assays, and gives our scientists also the opportunity to design the best possible experiment and then run it with the same set of equipment and protocols that they're used to. As an example, to build up this platform, here we have some data of colorectal organoids that were grown in 3D and subsequently treated with 5FU. But we've done this on 2 different liquid handlers that we have here in Leiden, but also with equipment in Utrecht. And we can see that we are getting rather high assay windows in all of these cases. Each assay gives us a very good set factor, so showing that we have a good screening window. And our CVs are low for the DimeZone negative control, so it's showing that we have consistent seeding and low variation in our treatment conditions. And then finally, with the concentration response curve, we actually see that the 5FU is giving us the same response in all of these assays, independent of the equipment or the protocol or the actual lab where the experiment was executed, and we're able to determine the same potency for 5FU in all these experiments. So it gives us quite some confidence that the platform itself is working stably, and that we can run this in different labs and across different equipment. Of course, then when we think about oncology, we're trying to find new drugs, new therapies. So does this also scale up? Well, here's a very clear example of how robust the platform is doing. So here we have a screening where we tested over 1,000 compounds in 2 colon and 2 ovarian cancer models, where the client was interested to find drugs that were targeting a particular pathway. And by using multiple models, we also cover different mutations within the diseases, and it allowed us to identify compounds for the client to progress into a next phase. And in this particular case, we used a multi-parametric approach, which is again one of the benefits of high content imaging data. You have so much information that you can use multi-parametric analyses. We used that to identify compounds that were not per se cytotoxic, which we classified as killing compounds here, but we were looking at compounds that still inhibited the growth and the development of these organoid models without inducing direct cytotoxicity, so that they were slightly more specific to the growth and that inhibition other than cytotoxicity. And just to show some of the data that shows the performance of the screen, and this is only the data for the colorectal organoids, is that we screened over 40 plates in these two models, and then in between all these plates we get rather stable and high set factor. Again, we have low CV, so low variation, and then when we look at the treatment data, we actually also get very consistent replicate performance. And while this may look like a quite standard type of visualization for high throughput screens and those kind of assays, it's also important to realize here that we've done this now with organoids. And in some cases, people still think that organoids are only a type of cell or type of model that is being used and grown in academic labs, that is very difficult or that it's not scalable. And that it's very difficult to get this to this type of level. But I think here we have very good data that shows that we're able to run this at a reasonable scale and that we actually get industry standards when we look at assay performance. And it basically showing that the organoids and organoid technology is ready to be used at screening levels and using those industry standards for assay performance and drug discovery. Of course, after the screening, our client was interested to make sure that the compound was actually targeting, was actually functioning through their specific target. So we created a knockdown model in the same colorectal organoid models. So after knocking down the specific target, we repeated the exposure with one of their main molecules. And indeed, what we can see is that in the knockdown, there's no longer a response, whereas in the non-targeting control and the wild-type control still show the same inhibition of growth with the treatment. So basically a rescue experiment showing that the compound that they had developed was specifically targeting the target of interest. And when we knock down that target, it's no longer functioning. In another nice example, we went from a primary screen to a preclinical candidate. We actually screened a large library of bispecific antibodies. This was done in 3 different organoid models in parallel. And we selected the most active bispecific antibodies and subsequently rescreened these in a larger panel of 30 colorectal cancer organoids to find out which of these bispecifics was the most potent or the most effective. And actually, when we start looking at the concentration response data, we will see a nice example here of that matched pairs of both the tumor model of the organoid and a normal model of the organoid. We see that cetuximab is equally potent in both types of models. But when we look at the bispecific antibody, we can actually see that it's specifically inducing toxicity or decrease in the organoid size in the tumor organoid model compared to the normal organoid model. So it's having a sensitivity towards the tumor model, but it's not affecting the healthy matched pair organoid. So this particular lead candidate entered the clinical trials early or in May in 2018. And I think it's currently still in development. But then also last year, we also published this story in Nature Cancer. And it's, I think, a very nice summary of what the technology with organoids and high content can give if you combine this in a well-designed study. This was a brief introduction into our organoid work. But of course, if we talk about complex 3D assays, there's still some things that we need to explore today. So on the left-hand side, we have those organoids. If we culture them again in 3D, we can actually make these assays more complex by turning them into co-cultures, which is one of our assay formats for immuno-oncology. So we can, of course, select the type of immune cells that we want to add to the co-culture. So this could be just PBMCs, it could be very specific populations, such as T cells, or we can even make triple co-cultures or other combinations of cells. And then on top of that, we can then add the therapeutic agents, either to stimulate or to induce particular effects in those immune cells. And then ultimately, again, use the high content imaging to then classify how the immune cells and the organoids or the 3D grown cells are interacting, and whether the treatment is actually inducing specific toxicity or killing or other type of biology. So one of those examples is shown here. So when we label the immune cells with the cell tracker and we add them on top of this 3D hydrogel, that we can see that over time the tumor cells are slowly migrating towards the tumors. Right, the immune cells are slowly migrating towards the tumor cells. And when we then look at the combination of a treatment, we can actually see that per specific treatments are able to induce the migration of these immune cells and thereby directing them towards the tumor cells. And then we can also analyze that by looking at the count and the location of the immune cells along the z-axis in the 3D culture. Then the next step, of course, is when we look at these immune cells infiltrating and targeting a 3D grown tumoroid or organoid, is then to classify or analyze what they're doing. So we often look at multiple time points. What we then often see is that at early time points, we can see that the cells, the immune cells, are infiltrating. So in this particular example, we have T cells that have been treated with a T cell engager. We can see that there is a clear response to the T cell engager that is actually inducing the infiltration at the early time point. And then if we look at a later time point, those T cells have not only infiltrated, but now have also had the time to start killing the tumor cells. We can see again that there's both a concentration-dependent effect of this T cell engager, but there's also a difference in the activity or the effect of these two different T cell engagers. And then indeed, what we can see is that with an increasing concentration, that we also see that increased toxicity happening in our images. And this is one of those things to consider with immuno-oncology assays, but I think also with some of our other assays. With our large biobank, it also allows us to select multiple models for screening or to determine the efficacy of some of your new drugs. It's also important to realize that indeed all these models are representative of the patient, so they will also have different characteristics and therefore also respond differently to therapies. So what we can see here with two different models in a standard IO setup with HLA matched PBMCs is that when we add the pre-activated PBMCs or naive PBMCs, is that they do respond differently, or we do see different degrees of killing between the two models. This is something that we see in our data with our models, but this is also representative of what we see in the population or in the patients, that not all treatments are working equally well. And for that particular reason, we quite often screen and run these assays in a panel of models to make sure that we capture those differences that we also observe in the patients. Then finally, when we talk about increasing the complexity, the EVPT platform, which stands for ex vivo patient tissue, is one of the most complex systems that we have, but it's also the most patient relevant because we're taking the fresh tumor samples directly from the patient. So within 24 hours, they're being processed and added to our assay plates for study. So this is taking the entire native tumor microenvironment from the patient as it is, with the endogenous immune cells that are still present and the other components that are there. They're all taken together and put into these 3D cultures. And then we can use that to test, again, how well potential new drugs are working. So in this case, we're so close to the patient. It's becoming a very complex platform because it is very dependent on the tumor samples that we get from the hospitals and our collaborators. Then we can only test a small number of conditions. So this is one of the last phases of studies that we can offer in vitro before you're moving into the clinical studies. And with the standardized platform that we have on the lab automation, we can just keep on running the same procedure, and again use high content imaging to look at the phenotypic effects of the potential treatment and how it's impacting the tumor, in this case directly from the patient. And then there's as an additional benefit to this platform is that we have more capabilities that we can do with it. So when we have these patient tissues, we can not only use them for screening and for testing new therapies in our 3D platform, but we can also use the tissue for further characterization using FACS and next-gen sequencing. And we can also still do a little bit of scale-up culture where we can use the further expanded models for additional FOX or RNA-seq analysis. So we are able to generate more data on the particular sample, which helps us better understand, again, the results from our 3D in vitro studies. I'm hoping that I've been able to show that using our organoid technology and high content imaging, so that when we combine it, that we've generated a very powerful tool for studying in vitro biology. So we're using the most physiologically relevant biology by doing 3D cultures and using the most representative type of models by having organoids and primary tissues available. And then if we combine that with the high content imaging, where we capture the full 3D structure and analyze all the individual cells and organoids that are available in that population, we're able to get a very clear idea and interpretation of how potential therapies are working and if are they inducing phenotypic effects in these tumors. And then lastly, I hope that I've been able to show you that by using lab automation, that we've been able to build a very stable platform that's allowing us to produce very consistent assays and also thereby generating very reproducible data that will thereby again support drug discovery. I would like to thank you for your attention and listening in, and I would like to open up the floor to any questions.
Thank you very much, Dr. Fokkelman. As with all the talks, we are likely going to have lots of questions from the audience, and I can see we have received some questions already, so I'll move straight on to the Q&A. Our first question is about the advantages of PDO and PDXO models compared to other methods for compound testing. Can you provide insights into this?
Excuse me, could you repeat the question?
Yes, so our first question is about the advantages of PDO and PDXO models compared to other methods for compound testing. Can you provide insights into this?
I think the most important reason and the most important benefit of using PDO and PDXO material is that it's far more physiologically relevant as a model because it uses the inherent mutations and genomic alterations from the patients for studying the effects of treatments on the disease and how the tumor effectively responds. If we look at cell lines, these have been engineered, immortalized, etc. So they're not very, they've just been around for so long that they've become reasonably artificial, to say, right? So the organoids are, of course, grown completely from the adult stem cell population. So they keep the genetic background, the mutational profile from the patient, and they keep those properties. So in that sense, it's just far more representative to the disease, the pathophysiology that we're trying to study.
Thank you. It would be great to get a practical insight into how organoids can be used for compound screening. Could you walk us through the process of screening hundreds of compounds in a primary screen using these models and highlight any key considerations?
Sure, yeah. It's quite similar to regular cell-based HTS or screening campaigns. Of course, you need to make sure that your assay is working well. But then I think the most important part is to make sure that you have expanded your organoids to a sufficient amount and that you can run the screening services with it. Right? You ideally want to run such a screen or such a campaign on the same batch of materials. And then, making sure that your lab equipment is up for the job, so you can run 10, 20, 40 plates that you need, and then test all the compounds that either you provide yourself or your client is providing. And of course, the other thing is to have the automation on your readout. So we also have indeed automated plate feeders for our imagers, so that you can just put a stack of 40 plates there, and then they will get imaged one by one automatically. I think for other considerations is mainly making sure that all your procedures are properly QC'd and that you know how they perform from plate to plate and run to run if needed. So all those procedures are still very important here. But then if you know how that works, then screening in organoids is quite similar to screening in cell lines.
Thank you. In the context of transduction of organoids, could you expand a little bit more on how you transduce the organoids?
It's actually quite similar to cell lines. You can just add the virus that you like to use to the organoid culture, and then basically wait. You can, depending on how you design your vectors, you can use selection, of course. Certain models do allow you also to go into single cell state. So then you could consider doing Fox sorting, but indeed there's quite a lot of models that do not survive well if you put them into single cells. So for those, indeed, using selection is a lot easier. You can of course do this with lentiviral, but we have also certain projects running with different viral or different virus types. So even virus screening is also very well feasible with organoids. So it's very straightforward actually. You just need to culture them similarly to a cell line, and then you have your transduced population.
That's great. Thank you for answering those questions. For your next question, could you tell one of our audience here if you're investigating the effects of cannabinoids on T cell stimulations against cancer?
Not specifically. So we are still a CRO, so a contract research organization. So we support small biotech up to large pharma in their research programs. And of course, we're helping them get the best drugs to the market, to the patients. So of course, I cannot disclose any compounds that they're working on. As far as I know, indeed, there's nobody specifically working on cannabinoids on T cells. I don't think we have something in that area specifically at this moment.
Thank you. You mentioned earlier on that each organoid model has unique growth characteristics and 3D morphology. Could you provide some examples of how these unique characteristics impact compound screening and drug development?
Yeah, so this is one of the things that we consider at the beginning of these projects as well. When we have all these models, we of course need to characterize them first, look how they grow, what their basic morphology is, and whether that's even representative for the disease or the study that you'd like to run. So I think in most cases we always have the nice clear set of pictures that show the differences in these morphologies. So like colorectal organoid can have clear cystic structures, but other organoids are more solid or more grape-like morphology. So they do have different requirements in terms of image analysis and how to quantify certain phenotypes. So that's how we mostly use that. And then, of course, the rest of the details can be addressed in the assay design and the study design to make sure that you find the compounds for a particular context that is most interesting to you. So you can always adjust certain media composition or adjust the assay setup if needed to create a different type of assay, right? But also considering things like a co-culture or triple co-culture. Those things are also feasible, but it really depends on the study design. And then we just customize the assay so that we get the best data for our clients.
Thank you, Dr. Fokkelman. We do have some time to answer one final question for this session. You mentioned the possibilities for preclinical validation in vivo follow-up experiments and direct testing in ex vivo patient-derived tumor material. Could you provide examples or case studies that demonstrated the effectiveness of these approaches in drug development and research?
So we have it's a very growing business, the EVPT platform for us. So we have several clients that are also like really recurrent clients that are working on new therapies, and they clearly see that this testing is helping them select the best candidates to move forward, right? So because the tissues are coming directly from the patients, they have a better understanding how well their new chemicals or their new treatments are working and which ones they potentially want to progress to still in vivo studies or already into clinical studies. But yeah, of course, some of this data is still with our clients, and in that sense, I'm not allowed to disclose what the particular targets or clients are. But I think there's also a few examples of this on our website, which is actually a very interesting resource for the listeners to look at. That we have several blog posts on these different platforms. So if you want to know more about the different morphology of organoids or the different capabilities that we have with high content imaging, and similarly for this preclinical testing with the EVPT platform, there are some nice examples on there as well that have more detail about the platform and what we can do with it.
Thank you so much, Dr. Fokkelman. Unfortunately, that's all we have time for today. And any questions that we didn't have time to get to will be sent to the speaker after the event. As with all the other talks in the event, this talk will now be available to view whenever you want for the next 30 days. That just leaves me to say thanks again, Dr. Fokkelman, and we'll be back soon with the next talk.